Medicinal chemistry. Drugs are classified in three main ways: by pharmacological effects, by chemical structure, or by target system. The classification can be challenging, however, because drugs may be structurally similar but have different pharmacological effects. Drugs are grouped depending on the biological effect they have. For example, analgesics, antipsychotics, antihypertensives, antiasthmatics, antibiotics, etc. This is useful if one wishes to know the full scope of drugs available for a certain ailment. However, it should be emphasized that such groupings contain a large and extremely varied assortment of drugs. This is because there is very rarely one single way of dealing with a problem such as pain or heart disease. There are many biological mechanisms that the medicinal chemist can target to get the desired effect, and so to expect all painkillers to look alike or to have common thread running through them is not realistic. A further point is that many drugs do not fit purely into one category or another, and some drugs may have more uses than just one. For example, a sedative might also have uses as an anticonvulsant. To pigeonhole a drug into one particular field and ignore other possible fields of action is folly. Classification of drugs by structure. Many drugs that have a common skeleton are grouped together. For example, penicillins shown here, barbiturates, opiates, steroids, catecholamines, etc. In some cases, like for penicillin, this is a useful classification since the biological activity is the same. However, there is a danger that one could be confused into thinking that all compounds of a certain chemical group have the same biological action. For example, barbiturates may look much alike and yet have completely different uses in medicine. The same holds true for steroids. It's also important to consider that many drugs can act at several sites of the body and have several pharmacological effects. The opiates are a group of drugs with similar chemical structures. These are compounds that are classed according to whether they affect a certain target system in the body, usually involving a neurotransmitter like antihistamines or cholinergics. This classification is a bit more specific than the first, since it's identifying a system with which the target interacts. It is, however, still a system with several stages, and so the same point can be made as before. For example, one would not expect all antihistamines to be similar compounds, since the system by which histamine is synthesized, released, interacts with its receptor, and is finally removed, can be attacked at all of these stages. What is a good drug? Can we draw a neat line between good drugs like penicillin and bad drugs like heroin? If so, how do we define what is meant by a good or a bad drug in the first place? The answers we would get would almost certainly depend on whom we were to ask. As far as the law is concerned, the dividing line is defined as black and white. As far as we are concerned, the questions are irrelevant. To try and divide drugs into two categories, safe or unsafe, good or bad, is futile, and could even be dangerous. First of all, let us consider the so-called good drugs, the medicines. How good are they? If a medicine is to be truly good, it would have to satisfy the following criteria. It would have to do what it's meant to do, have no side effects, be totally safe, and be easy to take. There is no pharmaceutical compound on the market today that can completely satisfy all these conditions. Admittedly, some come quite close to the ideal. Penicillin, for example, has been one of the most effective antibacterial agents ever discovered, and has also been one of the safest. Yet it too has its drawbacks. It has never been able to kill all known bacteria, and as the years have gone by, more and more bacterial strains have become resistant. Nor is penicillin totally safe. There are many examples of patients who show an allergic reaction to penicillin and are required to take an alternative antibacterial agent. While penicillin is a relatively safe drug, there are some medications that are distinctly dangerous. Morphine is one such example. It is an excellent analgesic, yet suffers from the serious side effects of tolerance, respiratory depression, and addiction. It can even kill if taken in excess. Barbiturates are also known to be dangerous. At Pearl Harbor, American casualties undergoing surgery were given barbiturates as general anesthetics. However, because of a poor understanding about how barbiturates are stored in the body, many patients received sudden and fatal overdoses. In fact, it is reputed that more casualties died at the hands of the anesthesiologists at Pearl Harbor than died of their wounds. To conclude, the good drugs are not as perfect as one might think. What about the bad drugs then? Is there anything good that can be said about them? Surely there's nothing we can say in defense of the highly addictive drug heroin? Well, let us look at the facts about heroin. Heroin is one of the best painkillers known to man. In fact, it was named heroin at the beginning of the 19th century because it was thought to be a heroic drug that would banish pain for good. The drug went on the market in 1898, but 5 years later, the true nature of heroin's addictive properties became evident, and the drug was withdrawn from general distribution. However, heroin is still used in medicine today, under strict control, of course. The drug is called Diamorphine, and it is the drug of choice when treating patients dying of cancer. Not only does diamorphine reduce pain to acceptable levels, it also produces a euphoric effect that helps to counter the depression faced by patients close to death. In fact, it's very structurally related to morphine, which has similar pharmacological effects. So how are we to define a drug in general? One definition could be to classify drugs as compounds that interact with a biological system to produce a biological response. This definition covers all the drugs we have discussed so far, but it goes further. There are chemicals that we take every day and which have a biological effect on us. What are some of these everyday drugs? One is contained in some teas, coffee, and cocoa. These beverages can contain the stimulant caffeine. Whenever you take a cup of caffeinated coffee, you are a drug user. We could go further. Whenever you crave a cup of coffee, you are a drug addict. Even kids are not immune. They get their caffeine shot from Coke or Pepsi or energy drinks. Regular use of caffeine leads to withdrawal symptoms that include headache and nausea. There can be little doubt that alcohol is a drug, and as such causes society more problems than all other drugs put together. One only has to study road accident statistics to appreciate that fact. It has been stated that if alcohol were discovered today, it would be restricted in exactly the same way as cocaine or cannabis. If one considers alcohol in a purely scientific way, it is a most unsatisfactory drug. As many will testify, it is notoriously difficult to judge the correct dose of alcohol required to gain the beneficial effect of happiness without drifting into the higher dose levels, which produce unwanted side effects. Alcohol is also unpredictable in its biological effect. Happiness or depression may result depending on the user's state of mind. Addiction and tolerance in certain individuals have negatively affected the lives of addicts and relatives alike. It may seem strange to include poisons and snake venoms as drugs, but they too react with a biological system and produce a biological effect. A bit extreme, perhaps, but a response all the same. The idea of poisons and venoms acting as drugs may not appear so strange if we consider penicillin. We have no problem in thinking of penicillin as a drug, but if we were to look closely at how penicillin works, then it is really a poison. It interacts with bacteria, the biological system, and kills them, the biological response. Fortunately for us, penicillin has no effect on human cells. For several thousand years, man has used herbs and potions as medicines, but it's only since the mid-19th century that serious efforts were made to isolate and purify the active principles of these remedies. Since then, a large variety of biologically active compounds have been obtained and their structures determined, like morphine from opium, cocaine from coca leaves, quinine from the bark of the cinchona tree. These natural products became the lead compounds for a major synthetic effort where chemists made literally thousands of analogs in an attempt to improve on what nature had provided. The vast majority of this work was carried out with no real design or reason, but out of the results came an appreciation of certain tactics that generally worked. A pattern for drug development evolved. Nowadays, the development of a novel drug from natural sources might follow the following pattern: screening of natural compounds for biological activity, isolation and purification of the active principle, determination of structure, structure-activity relationships, and synthesis of analogs. The screening of natural products became popular following the discovery of penicillin from a mold. Plants, fungi, and bacterial strains were collected from all around the world in an effort to find other metabolites with useful biological activities. This led in particular to an impressive arsenal of antibacterial agents. Screening of natural products from plant and microbial sources continues today in a never-ending quest to find new lead compounds. In recent years, organisms from marine sources have shown promise in yielding new drugs. Isolation and purification. The ease with which the active compound can be isolated and purified depends very much on the structure, stability, and quantity of the compound. Penicillin proved a difficult compound to isolate and purify. Although Fleming recognized the antibiotic qualities of penicillin and its remarkable non-toxic nature to man, he disregarded it as a useful drug since it appeared too unstable. He could isolate it in solution, but whenever he tried to remove the solvent, the drug was destroyed. Now we know the structure of penicillin. Its instability under the purification procedures of the day is understandable, and it was not until the development of a new procedure called freeze-drying that a successful isolation of penicillin was achieved. Other advances in isolation techniques have occurred since those days. There are now a variety of techniques available to help in the isolation and purification of a natural product. Structure determination. In the past, determining the structure of a new compound was a major hurdle to overcome. It's sometimes hard for present-day chemists to appreciate how difficult structure determinations were before the days of NMR and IR spectroscopy. Both of these approaches involve testing the compound to determine what the different pieces are that make up the chemical structure. These pieces are called functional groups because they confer particular properties to the compound, like interactions with other molecules and solubility. A novel structure that may now take a week's work to determine would have taken 2 or 3 decades of work in the past. For example, the microanalysis of cholesterol was carried out in 1888 to get its molecular formula, but its chemical structure was not fully established until an X-ray crystallographic study was carried out in 1932. Structures had to be degraded to simpler compounds, which were further degraded to recognizable fragments. From these scraps of evidence, possible structures were proposed, but the only sure way of proving the theory was to synthesize these structures and to compare their chemical and physical properties with those of the natural compound or its degradation products. Today, structure determination is a relatively straightforward process, and it is only when the natural product is obtained in minute quantities that a full synthesis is required to establish its structure. The fragmentation patterns can give useful clues about the structure, but it does not, however, prove the structure. A full synthesis is still required as final proof. Structure-activity relationships. Once the structure of a biologically active compound is known, the medicinal chemist is ready to move on to study the structure-activity relationships, SARs, of the compound. The aim of such a study is to discover which parts of the molecule are important to biological activity and which are not. The chemist makes a selected number of compounds which vary very slightly from the original molecule and studies what effect that has on the biological activity. One could imagine the drug as a chemical knight entering the depths of a forest, the body, in order to make battle with an unseen dragon, the body's affliction. The knight is armed with a large variety of weapons and armor, but since his battle with the dragon goes unseen, it's impossible to tell which weapons he used or whether his armor is essential to his survival. We only know of his success if he returns unscathed with the dragon slain. If the knight declines to reveal how he slew the dragon, then the only way to find out how he did it would be to remove some of his weapons and armor and to send him in against other dragons to see if he can still succeed. As far as a drug is concerned, the weapons and armor are the various functional groups present in the structure, which can bind to the receptor or enzyme or contribute to the solubility of the compound. We have to be able to recognize these functional groups and determine which ones are important. The interactions between the functional groups found in a drug molecule and those within each biologic target determine each property of the drug. The relative fit of each drug molecule with its target is a function of the number of physiochemical properties, including acid-base chemistry, functional group shape and size, and three-dimensional spatial orientation. The quality of this fit has a direct impact on the biological response produced. Similar to automobiles, refrigerators, and other machines, drug molecules consist of various components known as functional groups. This is exemplified by fluoxetine, Prozac, shown here, an antidepressant that selectively blocks the reuptake of serotonin in the brain. This drug is comprised of 7 parts or 7 specific functional groups. From a medicinal chemistry perspective, functional groups provide specific properties and behaviors that allow drug molecules to exert their desired pharmacodynamic and pharmacokinetic effects. For a given drug molecule, they play a significant role in the overall solubility, ability to interact with specific biological targets, specificity, mechanism of action, route and duration of metabolism and elimination, suitability for a specific therapeutic situation, and tendency to cause adverse effects or drug interactions. When examining drug molecules, there are 3 overriding concepts that you should always consider. First, every atom within the structure of a drug molecule is part of a specific functional group. Second, within any given drug molecule or class of drug molecules, some functional groups will be more important than others. Third, it is possible to alter functional groups to enhance activity, increase absorption, decrease adverse effects, or provide other therapeutic benefits. The elements of luck and inspiration. It is true to say that drug design is becoming more rational, but it has not yet eliminated the role of chance or the need for hard-working, mentally alert bench chemists. The vast majority of drugs still on the market were developed by a mixture of rational design, trial and error, and pure luck. The drugs that were achieved by purely rational design are severely limited. Frequently, the development of drugs has been based on ringing the changes, watching the literature to see what works on related compounds and what doesn't, then trying out similar alterations to one's own work. It is very much a case of groping in the dark, with the chemists asking whether adding a group at a certain position will have a steric effect, an electronic effect, or a bonding effect. Even when drug design is followed on rational lines, good fortune often has a role to play. Learning goals for medicinal chemistry. 1. Describe the ways that drugs can be classified. 2. Report the good and bad qualities of a drug. 3. Describe the purpose of chemically altering a compound structure. And 4. Understand the basic concept of functional groups.